Hey future AP Chemistry students, today we're going to be talking about AP Biology. So, let's start with the basics. What exactly is a catalyst and what does it do? A catalyst is a compound that can be added to a reaction to increase its rate. This process is called catalysis, and the catalyst is not used up during a reaction. This rate increase is achieved by providing an alternate pathway for the reaction, one with a lower activation energy than the original. But that's a very general explanation that doesn't quite explain the mechanics behind how this works. To get an idea behind the mechanics of a catalyst, we'll be looking at a catalyzed reaction between ethene and hydrogen gas. A catalyst needs to be reactive enough to temporarily break the bonds found within the reactants, but not so strong that those bonds become permanent. Atoms bonded to the catalyst migrate until a collision occurs, triggering a reaction and resulting in the formation of the product. This product is then able to break from the catalyst, finishing out the reaction. Now, not all catalysts work the same way, and there's practically limitless ways this could play out. I just need an example and decided to use this one. So, in summary, catalysts essentially mess with bonds in a variety of ways to provide an alternate low activation energy pathway to facilitate a reaction and increase its rate. If you want a fuller description of how they work, I've linked the University of Texas at Austin page I used to write up my script in the description. It's really useful and explains way more detail, so that I cut since they would have made this segment run on for way too long. Side note, if you have a reaction equation, you can identify a catalyst by simply checking which compound exists at both the beginning and end of a multi-step reaction. Now, I've been talking about activation energy quite a bit, and some of you may not know exactly what that is. You can always go watch a video on reaction energy diagrams that's probably somewhere else on Miss Nielsen's channel, but you decided to watch my video, so I'm going to explain it here. Strap in tight, this will be a long one. Activation energy is the energy required to kickstart a reaction. Everything before this curve's relative maximum, right here, and right here, is the reaction's activation energy. Now that we're done talking about the nerd stuff, it's time to talk about even more nerd stuff. I hope you're excited, it only gets worse from here. But Ryan, I already knew about catalysts. If you don't hurry up and get to the point, audience retention's gonna tank at this point. Dude, you gotta hurry! I literally do not care. Catalysts in biological systems are a special type of protein known as enzymes, usually named with the suffix ase, A-S-E. They function by accepting substrate, which we'll define as a synonym to reactants, into their active site, an indent formed by the molecule structure meant to fit a specific type of substrate, which can sometimes be specialized to a, specific molecule, like glucose or something. Once the active site is filled, the enzyme weakens and messes with the substrate's bonds to form the product, which then dissociates and releases from the active site. The enzyme can then take in another molecule, sticking to that idea of enzymes and catalysts not being used up in a reaction. So, what makes enzymes so important? It's simple. They lower activation energy and increase reaction rate. Take glycolysis as an example. It's a vital part of aerobic cellular respiration that uses a great deal of enzymes. Without this series of enzymes speeding up the reaction, there's no way we'd be generating enough energy to survive. Glycolysis would take obscenely long to produce pyruvate and require a ton of energy to occur. The process of cellular respiration would stagnate at step 1. Imagine how long it would take with the other steps factored in. Want another example of why enzymes are so important? Take a look at this study by University of North Carolina professor Richard Wolfenden and his co-author Charles Lewis. The reaction that produces hemoglobin, the vital oxygen carrier in our blood, would take a whopping 2.3 billion years without enzymes. This outshines the time span constructing DNA and RNA would take without enzymes, a speedy 78 million years compared to the glacial pace of enzymeless hemoglobin production. And so, that's about all I have on how catalysts function in biological systems. There's a lot of nuance to the study of how enzymes and all the related stuff works that I'm To put it quite simply, I'm not qualified to talk about. The only college level biology class I've taken is AP Biology, and that is absolutely not enough to talk about the field of biology accurately. So, unfortunately, I'm gonna have to leave you with this pretty much. But I highly encourage you to look into this on your own time. A lot of biology is extremely interesting. There's way more depth to it than you could ever dream of, really. I know I am interested in, I swear, every time I go into like Cell Bio or Nature.com or Science Director, any web archive of research, any web archive of research magazines and all that, I'm shocked at just the sheer volume of stuff that there is. Like, it's absolutely crazy. If you want to learn more about enzymes in organisms, specifically about their evolution and how we've learned to classify their functions, I have a few papers you might be interested in. The first one here is an overview on studies regarding the evolution and function of enzymes. It's a fun read and links to a lot of very interesting studies that'll take you down quite a fun rabbit hole if you have the time and interest. The other two are sources 25 and 26 of the previous paper, this paper, respectively. And both cover the possible evolution of enzymes in organisms. They're extremely interesting and are genuinely awesome reads if you're curious about the subject. And even if you're not curious, you might actually find them very, very interesting. A lot of the concepts in evolution are very cool, and it's fun to learn where a lot of the stuff found in our body come from. Anyhow, that about does it for this video. I hope you've learned something and have a better idea of how to connect the concepts you've learned in class to other fields and aspects of both science and life. Thanks for watching, and good luck out there, varsity chemists.